So we are in the wastewater surveillance lab. I'm the research project manager for the wastewater testing that we do here on campus. It started at the beginning of the COVID pandemic, and so we're monitoring wastewater routinely for the presence of not only SARS-CoV-2, but we've also added in other pathogens of interest, such as influenza, norovirus, polio, HMPXV or monkeypox. We had a massive team when we first started this. There was probably a dozen or so different people from all different disciplines and experiences. We met on Zoom and however we could, email and whatever, and that's how we kind of got rolling. And we got back in the lab and we made it work. We get samples from 3 different wastewater treatment facilities or 3 different sites. Campus sits right in the middle of town, and it is served by its own dedicated water treatment facility. Then we also have another site that sits kind of inside the community, which is the downtown area specifically. The surrounding community is served by a separate facility, which that has everything that is non-campus. So yeah, we're in a unique position that we can kind of look at, even in a very small space, we can look at very different community makeups.
So we're here at the University Park Water Reclamation Facility. Our role here is to basically receive all the wastewater generated on the University Park campus, receive that wastewater, and then treat that wastewater so that it's safe to be released back out in the environment. We have a flow meter that monitors how much flow we're getting from campus. On average, we'll get about 2 million gallons per day of wastewater that comes to us while class is in session during a typical fall or spring semester. Here's our composite sampler. This helps us take a representative sample of the incoming wastewater over a 24-hour period. And then that sample is collected in a refrigerated compartment with a sample collection container. So this holds the sample stable, that then we collect that sample and provide to the researchers for analysis of COVID.
So the wastewater sample is going to be vacuum filtered directly into another tube containing polyethylene glycol and also a little bit of salt. And what that does is once those compounds are resuspended in the sample is those are going to allow any viral particles or genetic material to be pulled out of solution. It kind of destabilizes those elements in the wastewater. So that's how we're actually able to concentrate the virus from a large 40 ml volume down into kind of a manageable size. So now these are ready to go on the centrifuge, so we can head upstairs and get these loaded. Now we need to get all those viral particles that are suspended in the solution, we need to pull them out of solution. And so that's what the centrifuge is doing. We're going to spin all these samples at 12,000 Gs for about 2 hours at 4 degrees. And what that's going to do is, again, viral particles are very small, very light. And so by applying all those G forces, that's going to cause them to pellet down at the bottom of the tube. Then we can just work with our concentrated pellet of hopefully virus and other things of interest. So you've heard with COVID-19, you take your PCR test, that it's the same process, we're just doing it slightly different. We're actually going to quantify or count how much virus is there. Whereas a lot of like the at-home tests, it's just a yes, it's there, no, it isn't. You don't actually know how much is there. I also think of it like you're taking an eye exam, you're at the eye doctor and they're having you read the chart. When the text is super, super small, you can't make out what letters they are. So what PCR in theory kind of does, it takes that text and makes it larger and larger and larger. And so it's like reading the top line on an eye chart where it's just one very clear letter. That's, in a way, what PCR is doing. This is going to go into our digital PCR system. The first thing that the instrument does, it applies pressure onto those spaces and it essentially injects the sample across the plate. And if I flip it over, each one of these rectangles that you see consists of 26,000 individual partitions. And so as the sample is kind of injected across that rectangle, all those individual reaction vessels, if you will, gets filled with the sample and prepped for the PCR test itself. So this is actually looking for SARS-CoV-2 virus. Each one of those illuminated dots, if you will, is a positive reaction. That is a reaction well that had virus in it. And so now the software, what it does, it takes a look at what was positive, what was negative, and using statistics, it can determine how many virus particles are actually present. So a lot of research, when done, isn't always maybe immediately applicable to the local community, but we are very fortunate with this research that it can be directly applied or used by the local community, local leaders, and that includes university health services here on campus. You know, when we start to see a rise in things, especially if it's been, 2-3 time points of a rise in virus, we can say, hey, things seem to be transmitting, we're detecting more and more virus, you might start seeing more and more cases start coming through your doors. And that's really, I think, a power of this surveillance system, is to kind of, even though we might not be able to say, hey, you're gonna have 10 people walk through your door at noon today, we can at least say, hey, you might see some people start coming through. And so far the reaction from the nursing staff has been incredibly positive, and thankful that, we're providing this for them. Beyond, the university as a whole, it goes on to the community leaders, so they can think about decisions that may influence, mandates, whether it's masking, distancing, whether they allow people in stores. We report out to the Pennsylvania Department of Health. They also see our numbers, and they have their own statewide dashboard, and even beyond that at the national level. So our data is reported into the CDC's National Wastewater Surveillance System as well. So we are really from the very small scale of the university level all the way up to the national level. So it's just empowering the local community to potentially make better decisions for themselves. The more information that they have, the better decisions they may be able to make. And I think we really empower them to do that.